All right, so I debated whether or not to include this lecture as the last kind of capstone to our plant lectures. But I think it's so interesting and it involves a few experiments, and so we're going to go over it. This will be kind of the capstone to our plants. All right, so plant behavior is kind of a little more, I should say, basic than when looking at animal behavior, because we're not looking at how they run, walk, or swim, or touch each other or anything like that. But plants still can change their internal composition in response to stimuli. It may be in response to damage from herbivores or physical damage from storms. They can also grow in response to nutrients or sunlight, or even in response to the shading of other plants. So the regulation of their behaviors occurs through plant hormones. And a hormone is a signaling molecule, so something that originates from one tissue and then travels through the plant to a different part of the tissue, the target tissue. And this causes a molecular change, or causes a cascade of events which initiates a response. These are different types of hormones: gibberellins, auxins, cytokinins, ethylene, abscisic acid is what ABA stands for, and they control a variety of plant behaviors. One of these plant behaviors includes what's called phototropism, which is basically a response in the form of movement towards or away from light. Tropism then refers to the curving towards or away. An example of this is the sunflower. The large heads of the sunflower, which form the flowers themselves, will rotate with the sun. So with the rising of the sun, they will be pointed towards the east. They will follow it across the sky and then be pointed towards the west as it sets. So many experiments have been shown to reveal the nature of hormones in plants, and we're going to go over a few of those, starting with Francis and Charles Darwin. So Darwin was not just, although he's most well known for his theory of evolution, he did a lot of other experiments as well. And I believe Francis was his son. So what they did is they have these sprouting from leaves, these little plants, which will eventually grow into more mature plants, but they're called coleoptiles. Okay, so this is a little coleoptile here. And what they observed was that they directed themselves towards light. They would bend towards light. And so the Darwins put different caps and sheaths on these to see if it would affect how the light, or how it would respond to light. So they put an opaque cap on the top, and they found that it did not bend towards the light. They put a transparent cap and found that it did bend towards the light. Okay, so just the physical nature of the light was not what caused it to bend. And then they put an opaque shield over the curvature to see if that affected it, and it still bent. So the conclusion was that the light on the tip caused the phototropism. It doesn't matter what was happening towards the middle and bottom of the plant, but at the top you had something causing the plant to bend towards the light. So a follow-up experiment to this was done by Boyson and Jensen. And what they did is they cut a tip, and then they put a gelatin in between the tip and the rest of the coleoptile. And they used two different types, an impermeable gelatin and a permeable gelatin. And what they found was the impermeable gelatin, even though everything else was intact, it didn't allow things to flow from the tip to the middle part of the coleoptile, and it did not bend towards the light. But a permeable gelatin, which allowed chemical substances to flow through it, did. Okay, so the conclusion is that there's something coming from the tip to the site of the curvature, causing it to curve. All right, finally, Wint did a number of experiments with agar gel, which absorbed chemicals placed under the tip. So he exposed tips to dark and then cut them in cubes. Okay. And then when placed on the coleoptiles, so here is this tip, he cut it off and he put it on this agar so it could absorb whatever was in it. And then he cut these little, cut the augers into different tips and put them on coleoptiles that had their tips removed. And so when it was put on there, it bent away. So the conclusion was the chemical on the dark side of the tip caused the elongation of the coleoptile. Okay, so this was where the elongation was occurring with the transfer of whatever was being submitted by the tip. And he named this chemical auxin. All right, so the way that this works is through a bunch of other experiments. We've figured out the mechanism by which it works. First, auxin causes a proton pump to be activated and protons to be pumped out of the cell and into the extracellular space where the cell wall is. What the protons do is they increase the concentration of hydrogen ions, which decreases the pH. Okay, so it causes it to be more acidic. And this acidic increase in acidity causes this enzyme called expansin, so that's what this enzyme is here, to be activated, and it then cuts the cross links between the cellulose microfibrils. So normally you have all these cellulose running throughout the cell wall, and then these cross links tying them together to hold everything together. And then expansin will go in and cut those cross links. And then what happens is the turgor pressure from having that hypertonic solution in the inside the cell will then cause the cell to expand, and thus the cell elongates. And when you have all those on one side elongating, then it will cause the coleoptile to bend. All right, so this is the schematic from your book. Again, auxin causes proton pump to be activated. Protons are pumped to the outside outside of the plasma membrane into where the cell wall is. That activates this enzyme, expansin. Expansin then cuts the cross links, and then the turgor pressure causes the cell to expand. So that is just one thing that auxins do. They also are used, they have many other types of cell behavior that they also control. They're sometimes used as pesticides to disrupt the life cycle of the plants. Other hormones, including auxins, are signals for apical dominance, apoptosis, which is cell death, cell differentiation, germination, fruit ripening, senescence, which is stopping reproductive efforts, and abscission, which is the shedding of leaves. And you can see on this chart the different hormones and what they affect. All right, so one of these I just wanted to talk about again is ethylene, and that wasn't mentioned on that list. This is the aging hormone in plants, and it is secreted as a gas. And it is also called the ripening hormone because many fruits rely on this signal for them to start ripening. So apples and pears and tomatoes are an example of that. It is also used commercially to ripen tomatoes, bananas, pears after harvesting. The idea is you can pick unripened fruit when it's green, and when it's harder and more robust, and then you can transport it. And then once you get it to where it needs to be, you can ripen it by exposing it to ethylene. You can even, with what this schematic is showing, you can even take out the gene for ethylene so that they can't make their own ethylene, they can't ripen by themselves, and that even makes it even easier to transport them as unripe fruit. And then once they get to where they need to be, you can apply ethylene so that they ripen. All right, that's it.